Hello, everyone, and welcome to today's webinar titled Optimizing dsRNA Removal in IVT mRNA, Strategies for Enhanced Translation and Reduced Immunogenicity. This webinar is a part of the second annual Cell and Gene Therapy virtual event series. I'm Melanie Chen of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by CELLSCRIPT. For more information about CELLSCRIPT, go to www.cellscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. I now present today's speaker, Johnathan Parker, Senior R&D Scientist at CELLSCRIPT. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Johnathan, you may now begin your presentation.
Awesome. Thank you so much. So we're going to talk about mRNA production a little bit, the dream and the reality of it. We're going to talk about double-stranded RNA. We're going to talk about kind of defining our quality standards by the type of RNA that we're making and the use that it is intended for. We'll talk about strategies both in the industry at large as well as internal work to reduce double-stranded RNA as a means of improving translation and kind of the consistency and usability of a lot of these different tactics and how useful they may be to any customer. So back to basics is the ideal mRNA structure that everybody wants out of every single reaction that they get, which is fairly straightforward. Everyone's looking for, in terms of mRNA production, a cap structure, preferably cap 1, where you have your 7-methylguanosine of 5-to-5 triphosphate bridge, and then a base +1 with a 2'-O-methyl with some variation, but it's really the meat of mRNA. And then you'll have a poly A tail on the other end, which can be varying lengths and encoded or enzymatic. Those two together, cap plus tail, make a very, very effective translational complex. And then you'll have your coding region, which typical industry standard right now is to have a lot of base substitution with either pseudouridine or N1-methyl pseudouridine, which really goes into pretty much every mRNA reaction just for the purposes of reducing immunogenicity. Now, this is what you want. You want 100% of this, and that is sort of the dream of RNA production. Now, the reality is a little different. While you can go through an IVT reaction and hopefully get full-length transcript, everything that you want, what you may end up getting is a plethora of other byproducts. One of those is premature termination, where the T7 polymerase will just fall off during IVT. Sometimes it just runs out of nucleotides, can't find them. You'll get long double-stranded RNA. We'll talk a little bit about that later. You'll have your DNA template that you used for producing that full-length transcript. That's just an obvious contaminant, can't get away from it. You'll have short dsRNA, which has different mechanisms of being produced than long double-stranded RNA, but we'll talk about that too. Abortive transcripts, which usually are very short, about 20 nucleotides just from the T7 polymerase trying to get started. And then you'll have random small RNA, which can be there in so many different amounts, and those can be sense or antisense, and they'll form from loops in your RNA that form, and then T7 just starts churning around and creating more stuff. Now, none of these things are really novel. We've known about them for a long, long time. It's just that when you actually make RNA, it's not a clean, homogeneous process usually. Now, for the most part, all these products are not cause for alarm. shouldn't be looked at as being some sort of a really, really bad thing. They're kind of just the name of the game. For instance, premature termination and random small RNA and abortive transcripts, they're either going to be so small that they can be purified away easily, or because they are really just the sequence that you wanted, maybe just an incomplete version of it, they shouldn't be immunogenic. They should be clean sequences that don't cause any real problems. Even if they do persist, it's really not that big of a deal. The DNA template as well, it's very easy to degrade and purify away. So DNA template is never considered to be that much of an alarm. It's just a product. That kind of just leaves us with the double-stranded RNA, whether it's short or long. And it has the disadvantage of both being problematic from an immunogenic standpoint, which we will talk about, as well as being very persistent. It's hard to get rid of using just classic precipitation or any sort of garden variety lab purification techniques. And that's been really at the root of trying to come up with interesting strategies for getting rid of it, which can be simple, they can be complex, but it has become a focus of the industry at large to remove that as much as possible, because mRNA's final use is really in living systems, and we'll talk about why that is not good for living systems. The formation of double-stranded RNA is interesting because it uses a lot of different classifications of what RNA is and what T7 does to be created. And the most interesting and probably common type of double-stranded RNA is something called turnaround transcription. Now you have your DNA template, right? And you have your RNA that gets created on that template. But because of the unique aspects of the T7 polymerase, it can sometimes just keep going when it reaches the end, which is shown by that little yellow arrow there. And if it's complementary enough to the RNA that you just created, it will sit down on that RNA and then keep moving along, because T7 polymerase, the wild type, has an ability to associate with RNA molecules. This one is pretty common, but we also have other ones that have completely different mechanisms of double-stranded RNA production. Random priming by small RNA is a big one. Because there's so much of those byproducts being formed, this one is especially devious. You'll have your normal RNA strand that you like, but then you'll have all that small stuff that, if it's so small, it might be complementary to something. There's a good chance. And then when it sits onto your transcribed RNA strand, the T7 can come along and use it as a launch point for creating double-stranded RNA. This typically makes small double-stranded RNA, as opposed to the turnaround transcription, which can make very, very large double-stranded RNA. The third type, which is probably one of the a little bit less common and a little bit more, I would say, unstable in terms of whether it's formed or not, because it depends on a lot of different factors. It's called antisense transcription. This one is also starts with the DNA template strand, where you have a transcribed RNA. It's good stuff, the stuff that you like. But then your DNA template strand has a non-template strand that is complementary to it. And because T7 has a tendency to just use whatever it finds, it can actually just sit down on that and create fully complementary RNA. And then all that happens is those two strands come together and form pretty big pieces of double-stranded RNA. Now, broadly speaking, these two things are put into categories of either being RNA or DNA dependent. Now, that's important because the ability for T7 polymerase to associate with RNA is a big target for ways to reduce double-stranded RNA that we'll talk about a little later. Now, going back to our previous slide talking about double-stranded RNA, as well as all these other products and why double-stranded RNA may not be good for living systems, there's a whole slew of different genes that double-stranded RNA tends to either cause the expression of or immune sensor mechanisms within cells that it tends to trigger. The reason is very simple. Double-stranded RNA doesn't exist in the world. It's usually viral. In living systems and eukaryotic systems, it's usually shown to be that a virus has infected the cell, and things are not going to progress well unless something is done about that double-stranded RNA. In terms of what cells do, this is even a simplification of all the different double-stranded RNA sensors that are out there, but these are kind of the heavy hitters. Now, one of them is pretty well known to just start degrading kind of any RNA that it finds, even if it starts with a double-stranded that it hit, it will start to attack all RNA that it comes across, which is not good because cells need RNA to make protein and protein to live. Some of the others will start to do translational arrest and apoptosis. Now, slightly different, but kind of the same outcome. Instead of degrading the RNA outright, it will just cease production of proteins in general, which tend to lead to apoptosis very quickly. And sometimes they will jump straight to apoptosis because if there is a viral infection, it's just better to kill the cell. And a lot of them really just lead to interferon and cytokine production. The crux of that is that interferon and cytokine production then just lead back to translational arrest and apoptosis. Interferons and cytokines produce this state that the virus is in a cell and that the cell is sick and that the living system in the general area is sick. And so it begins to sort of take up arms as a way of preventing that viral infection from going further. And every time you do a dose of, let's say, dirty RNA that has a bunch of double-stranded RNA in it, it's going to get worse and worse, sort of like an allergy getting worse and worse every time you are exposed to it. Same concept. It will build up and get worse and worse. If it doesn't kill it outright, it will usually kill cells, for instance, in future doses. Now, that's not to say that it's all doom and gloom for every single system. The quality tolerance for what your RNA is used for matters immensely. For instance, at the top of this upside down pyramid, enzymatics and normal R&D work, where you're just testing whether some enzyme works well on some RNA or mRNA or other nucleic acids. If you're producing mRNA to do this, it probably doesn't matter. Double-stranded RNA won't really affect in vitro systems that badly. There is always the potential that it will, but it's not a complex system. You don't have all those immune sensor genes, and so it won't really matter as much by the numbers. Then you move down to cell culture and gene expression. Well, depending on stuff like the type of cell, the type of gene, and how many doses it's getting, and what sort of cells they are, really is kind of the biggest determining factor here. Maybe what you'll start to have is some of the effects of reduced protein expression, but the cells themselves will be able to tolerate one dose, a few doses, without too much ill effect. You go down further and further, cell reprogramming, vaccine development, protein deficiency, cell and gene therapy. Each one of these sort of increases the complexity of the living system that it's in. Cell reprogramming is very sensitive to stuff like double-stranded RNA because it requires exact dosages. It requires everything to be very exact. And when the cells are actively fighting what they view to be a viral infection while trying to be reprogrammed for a specific cell type, it does not work out very well. Sometimes it makes things difficult. Sometimes it makes things impossible. Vaccine development's an interesting one. It can tolerate a little bit of immune response because that is sort of what you're trying to do with a vaccine. But protein deficiency treatment, where you're trying to supplement a protein into a person, or cell and gene therapy, you need everything to be exact and work every single time, which means having no double-stranded RNA would be kind of what you would want. And since that's sort of where a lot of biotech tries to end up, which is therapeutics, that is sort of kind of the the gold standard that we think of when we apply mRNA for anything. So prevention's always nice. It'd be nice to prevent it rather than to fix it after the fact. And in that light, preventing dsRNA has become a big aim of the industry for the last probably 5+ years, even more than that in terms of development probably. But T7 RNA polymerase, protein conformations and behavior have been extensively studied because people want to try to figure out where this short and long double-stranded RNA is coming from, and how can we adjust the T7 RNA polymerase structure in order to just try to prevent it, whether prevent it entirely or simply reduce it. I think it's more likely that you're just going to shift the numbers and try to reduce it a little bit more. And so with the information that we have about T7 RNA polymerase, people have tried extensive mutational studies to try to produce the best possible mutants of T7 to produce the least amount of double-stranded RNA while still producing the same amount of high full-length RNA. And this particular paper was pulled from Moderna, who was one of the larger companies that had done mutational analysis and found a couple of gene locations that it may work to help reduce it in. And then as an interesting side note, just to show the variability in T7 RNA polymerase, there was a Shanghai-based group that recently performed DNA shuffling to produce their own mutants. Like Moderna, they had the same intent where they wanted to reduce T7 RNA polymerase's ability to associate with RNA. And if you reduce the T7 abilities to associate with RNA, then that's really the main source of double-stranded RNA. However, unlike Moderna and unlike a lot of other companies that have tried mutational analysis, they tried to throw in some extra stuff for reducing that DNA-dependent double-stranded RNA formation that I talked about, which is not the most common one, but it is probably going to be one of the most persistent one. This one was interesting just because they targeted 3 completely different gene regions that had been targeted before, and they did a big triple mutation. So T7 RNA polymerase is a large protein. There's a lot of stuff to be done. The problem with doing these sorts of studies is you need a lot of resources to actually produce effective mutants. And you can't just use a lot of the ones that are on the market right now because they are protected, even if they are known. And even the reduction of double-stranded RNA has been proven through a lot of these mutants. It doesn't reduce double-stranded RNA to 0. It reduces it to a lower level. And how low that level is changes quite frequently, as I think we'll show. Now, if you can't prevent it, let's say that you don't have the resources to produce a bunch of T7 mutants, or the reaction condition optimization doesn't work for you, there's a lot of other strategies for downstream purification after the fact. There's a very interesting Repligen paper by Nathaniel Clark that did a lot of focus on all the different chromatographic methods for removing double-stranded RNA. His specific company focused on affinity chromatography, but they did a lot of comparison with cellulose chromatography with reverse phase HPLC, which I liked because it was able to take the same RNA and try to apply multiple different criterion to it. And so they showed, which aligns with a lot of data digging that you can do and see with how these guys perform in the industry, that cellulose, which is probably the most common one used right now because it's cheap and it's easy, and it tends to work well enough, reduces double-stranded RNA to a certain level. But if you want to get below that level, you have to go through a more complicated process like HPLC or affinity chromatography, which can reduce the double-stranded RNA quite a bit more, order of magnitude or a couple order of magnitudes more than cellulose. The problem with that is that the percent recovery of those reactions will go down. Now, if you spend all this time making mRNA, it would be rather unfortunate to lose about half of your RNA by putting it through HPLC, or 20% through affinity chromatography. It is better, and so perhaps that would make it a little bit more attractive than HPLC. They each have their pros and cons. They reduce it. They don't eliminate it. You lose a lot of RNA, but that is another option if you don't want to perform stuff like upfront T7 mutational analysis, which is very resource intensive. This is very, very material intensive. In addition, that affinity chromatography stuff was sort of interesting in what it showed, which was that when they did a standard versus an optimized IVT reaction, they found that affinity chromatography works even better when there's less double-stranded RNA in the first place, which I guess is an interesting result. Although all of their data was great, and they showed that the amount of interferon that was produced from this purified double-stranded RNA was lower. The improvement in GFP production, cells were healthier and happier for longer. It was just interesting that they found that you need to already have low double-stranded RNA in order to have the results work even better. And so that's sort of the current industry. The industry says that if you wanted to eliminate double-stranded RNA, you either do an upfront mutational solution or you do a downstream chromatographic solution. And they are nice in their own ways, because if you have a mutant T7, it's simple and low cost. The problem is always getting the mutant T7. And then the problem after that is having a good enough mutant T7 that you now don't have to worry about double-stranded RNA anymore. Problem is that if it doesn't quite work because of the conditions, or maybe the mutant doesn't give you as low as you need, then you still have to take it through downstream purification. And you sort of lost the advantage of having that mutant T7. So that lower limit still may be too high. You just won't know until you test it. And then the downstream purification by chromatography is nice because it doesn't really matter necessarily all the time what you're getting up front. You can remove it using chromatography. But even the simpler and cheaper methods for chromatography still require specialized equipment. They can require more personnel and protocols. The bigger the scale, the bigger the equipment that you need. And then the more complex methods, which can be, some of the best resolution and give you the lowest amount of double-stranded RNA, come at a different cost, which is that you tend to lose a lot of RNA, which is not great. And so that's leads us to our kind of alternative for these guys, which is an enzymatic approach, because it may provide a nice balance between a more complex method and a really well-performing method, not really having some of the limitations of either. And so some of the internal data that we've developed as part of our app notes that we'll release, we quantified that the effect that double-stranded RNA and nucleotide composition will have on the performance of, in this case, a luciferase mRNA transfected into monocytes. Now, we chose monocytes for a very specific reason, because monocytes are very sensitive to immune response. And if there's even a little bit of double-stranded RNA, the hope and the theory is that they will be quite unhappy, which is good because it allows us a very conservative way to measure double-stranded RNA's effects on the end goal, which is how happy are the cells, how happy is the living system. The experiment was fairly straightforward. We either did cells or mRNA with UTP or N1-methyl pseudo-UTP, and then each one of those was either purified or not purified with dsRNA removal using CELLSCRIPT's Immune Gold dsRNA removal kit. To the right here, we show capping and tailing metrics for all the RNAs that we tested using our ECQC kits, and everything was very comparable, which was nice, because having mRNA quality metrics to actually show that everything is above board is nice. So first and foremost, when we're talking about doing some sort of a translational study, you want to make sure that the amount of protein that is being created from that study is comparable or not. In this case, we were testing luciferase. So when we looked at the average RLU of the luciferase assay from all of the different mRNAs that we transfected into monocytes, we saw that all the way to the left with no RNA, you get no response. Makes total sense. With the canonical RNA, that it was not purified, meaning it just had UTP and the full IVT amounts of double-stranded RNA, you get a specific response. Take that one step further to the very middle column, and you add double-stranded RNA removal to canonical NTPs, and all of a sudden it jumps up. In this case, it jumped up to be about 9x of what the canonical one was. Take it a little step further, you can either add N1-methyl pseudo UTP in place of UTP, or combine N1-methyl pseudo with double-stranded RNA removal, which both sort of maxed out the luciferase assay. The one good part about combining both protocols together from a strictly luciferase assay perspective was the fact that it had a much more narrow standard deviation, which was nice. Repeatability is generally pretty good, but those two results ended up being about 10x what the canonical results were. Now, since monocytes are so sensitive to immunostimulation, all this stuff makes a lot of sense. N1-methyl pseudo UTP and double-stranded RNA are each implicated in causing some sort of an immune response. And so both of them are being likely mitigated in this data to produce a much, much better protein expression level. But we took it one step further. We wanted to measure what the immune response was from these cells. And to that effect, we measured 7 different gene expression levels for immune sensor genes using RT-qPCR, using targets for toll-like receptors, tumor necrosis factor, retinoic acid inducible gene, oligoadenylate synthetase 1, melanoma differentiation associated gene, and protein kinase R. These are sort of some of the heavy hitter gene immunostimulation sensors, and if they look good or they look get bad, it's a very good indicator of whether your RNA is good or bad. And in this case, I think that the overarching message, which I will show in numerical format in a second, is that without any RNA, everything was pretty low, which would be the blue bar on the left of each one of these. Using canonical nucleotides with double-stranded RNA, the orange bar, you see an immediate spike of a lot of different immunostimulation. And then depending on different levels or different types of either dsRNA removal or N1-methyl pseudo-U, which would be green and blue, you see a reduction. But interestingly, and I'll talk about this in a second, you see different reductions in different genes depending on whether dsRNA removal or N1-methyl pseudo is performed. And when you combine them together, N1-methyl pseudo UTP and dsRNA removal, you get the lowest levels of every single category, even usually less of an immune response, even compared to the no RNA control. So looking at these immune sensor genes is interesting because you can see that there is a bit of segregation on the intensity and the type. And there's a very good reason for that. If you look at the bottom block, the RIG1 through PKR, these are all dsRNA specific immunogenicity sensor genes. And with just dsRNA removal, you reduce all of them by at least 70% and up to over 90%. As opposed to N1-methyl pseudo UTP, which sort of has a more broad reduction immunogenicity, which I think is why N1-methyl pseudo-UTP was so attractive in the first place, is it just helped reduce immunogenicity, but it doesn't help nearly as much with dsRNA specific immunogenicity genes usually. When you combine both of them together, you get a really huge difference. Every single one of these immunogenicity markers is reduced by either 63 or up to almost 99%. And so you get a really, really clean and really, really low immunogenicity setup for cells. But there is a bit of segregation, and it really depends on what is it that you're trying to target. Reducing general immunogenicity moderately, or reducing general and specific immunogenicity very, very strongly. It's important. Taking that one step further, we, in addition to all of those specific genes, we also measured interferon beta using RT-qPCR. And across the board, I think it follows a lot of the previous results, where if you have canonical nucleotides, and I couldn't even show these using a normal bar graph. I had to use log because the difference is just that huge, where the normalized expression of interferon beta in the canonical nucleotides is 89.54, and that's reduced by almost 2 orders of magnitude whenever you introduce dsRNA removal into those systems. And also N1-methyl pseudo-UTP helps a bunch, just not quite as much as dsRNA removal. And as we've talked about, a lot of the dsRNA sensor genes, interferon is its main mode of action, where it's creating that antiviral state in cells. And so it's really cool to see how strongly that can be reduced by just removing double-stranded RNA. Yeah, and so the idea, which is interesting, is that each one of these tactics, whether you want to add N1-methyl pseudo-UTP, do some nucleotide substitutions instead of the normal UTP, and dsRNA removal can both act on really, really interesting parts of the immune system, and can each reduce immunogenicity of a system, but combining the both of them together can drastically reduce immunogenicity and also create kind of the best possible outcome for protein expression that is targeting pretty much every single aspect of what could go wrong in a cell from an impure mRNA standpoint. So that was a really, really cool result. And seeing as how N1-methyl pseudo-U is a fairly standard approach, tacking something onto it and seeing sizable gains was quite nice. It is also nice to see the protein expression seems to top out eventually. So the gains are really had by reducing immunogenicity, which will come into play as you do further doses down the road. So I think we want to think about the long-term sustainable kind of use of these tactics for creating the best possible mRNA. And I think that the overall idea of double-stranded RNA is that it's always present at some level. You can kind of try and struggle to make clean samples using prevention, in this case, in the upstream reaction, but it's always going to be there. Now, how much is there is really what everyone is trying to figure out. That bar table on the right showing that depending on the type of transcript and the type of nucleotide that you use, whether you're using UTP or pseudo or N1-methyl pseudo, and a bunch of different, luciferase or EGFP or SOX2. I mean, there's almost 2 orders of magnitude difference between some of these samples and how much double-stranded RNA is produced from them. So, reaction condition optimization is something that's been around for a long time to try to improve double-stranded RNA with the lowest cost easiest method, which is to just intelligently design your transcripts. But the problem is it's not super consistent. Even if you were to come up with a really good method, you're still going to go above that safe limit of double-stranded RNA once in a while, at the very best scenarios. And then on the flip side, you have the mutant T7 RNA polymerases in this graph on the left. Now, when you compare it to the wild type, which is the furthest left bar at 0.14% double-stranded RNA, it's really, really nice to see that you're getting a reduction in some of those other mutants, right? You know, you're getting probably 60 to 80% reduction. And so overall, that looks and feels like it's a good result and a very attractive option. But here's the rub, that even at that level that you see with the T7 RNA polymerase mutants, there will still be enough double-stranded RNA within those samples to produce quite a substantial interferon response. Now, this is even for simple applications like gene expression in sensitive cells for monocytes. Perhaps, you know, cell reprogramming won't even work with some of these mutants. And then protein replacement therapy and CAR T therapy may be endangered. In a therapeutic sense, they may not work, but it may also be just damaging to the person that it's used. And so you really have to be careful how you interpret some of these big reductions in double-stranded RNA by what you're about to use them for. In this specific case, if I were to, and if everyone at CELLSCRIPT were to look at some of these data and say, is the sample clean or not for double-stranded RNA, it wouldn't even come close. Our internal spec for double-stranded RNA contamination is less than 0.005%, of which most of these are about 400% too much. So how do we ensure that that is all brought down to that level? As I previously talked about in the immune sensor paper with cleaning up dsRNA internally, we bring everything down below 0.005% using our MinImmune Gold kit for double-stranded RNA removal. Now, it works well, but more specifically, it works well for our scale and for our aim. You know, we're an enzyme company, and so we would like to be able to make kind of the best quality mRNA without chromatographic methods, without additional equipment, without additional personnel. And so this really kind of checks all of our boxes in terms of what a good method is. And the best part, and something that I talked about earlier with chromatographic methods, is that they tend to work a lot better when there's already less double-stranded RNA in them, because most systems that are like chromatography will reduce it by a percentage amount, a certain fold reduction. But what if you had upwards of several percentage of double-stranded RNA? Would your after product be able to meet that kind of criterion? It's a consideration. You want something that will work across a bunch of different starting double-stranded RNA concentrations. On this graph on the right, we have 4 different IVTs that were produced using either CELLSCRIPT's IVT kit or 3 competitor kits. And you can see that there's quite a large variation in the amount of double-stranded RNA that will be produced, from 0.045% to almost 0.4%. So pretty large discrepancy, or whole order of magnitude, just in the baseline amount of double-stranded RNA. But directly next to all those bars, you have that exact same sample that's been treated with the CELLSCRIPT's MinImmune Gold dsRNA removal, and all of them are brought down to less than 0.005%. In fact, if you'll notice, they're all brought down to pretty much the same level, which is our limit of detection. And so that 0.005% as a spec, just to provide some amount of background information, it is really close to the lowest that we can reliably measure using immunoblot. If we were to look at it from a realistic perspective, it's actually quite a bit lower than 0.005%. It's not just at 0.005%, it's just too low to detect. And so this is really important to us to as often as possible and for every single RNA that we can possibly do it on, to reduce double-stranded RNA low so that it could be used for everything up to CAR T cell therapy. There's some really good papers that have actually used MinImmune Gold-like protocols to reduce double-stranded RNA for CAR T cell therapy, and it was a huge difference even when compared to stuff like base substitution. So it's important to keep stuff at this super low level just for stuff like cell reprogramming success, and anything more complicated than that. And also at this level, just if we're looking from an actual numbers perspective, at this level, interferon production is essentially going to be zero, which is also very important because even if you had a successful performance of a single batch or dosage of mRNA, you have no idea of knowing how badly that may perform as you continue to do future doses, whereas we can do cell reprogramming, many doses with interferon on cells that are even not super optimal, and it'll work well. And so in this case, I do want to bring attention to that table there, where of course it depends on what your starting amount is, the percent difference, but we're seeing either 93 to 99% reduction in double-stranded RNA, and it's all being brought down to the same level. Doesn't matter how much it starts at. And so the real secret to it is that we've created an optimized protocol, we've created an optimized enzyme and an optimized buffer to ensure minimal nicking from MinImmune Gold kit. We have plenty of disclaimers that we talk about, and we want to make sure that everyone knows that if you have a super highly structured mRNA, you want to test it on a little tube first to make sure everything is above board. But it's very easy feasibility to be done. Doesn't require more than a couple hours just to know if it works fine. But it's totally scalable. It can benefit from tangential flow filtration, just like bulk IVT, which is done in huge scales. It's the same thing because an enzymatic solution doesn't require any special material. It just requires RNA. It requires the enzyme. And the percent recovery is great too, because it's only going to be limited by the purification steps that you already use. If your purification step has a really high recovery, then this will have a really high recovery because it's just the same as what you use to produce IVT with, I assume. In this case, I grabbed from for these two tables and gel images at the bottom, I just grabbed a bunch of different RNAs that we use for anything in the lab just to try them and see if it worked. And it was as simple as trying it with and without RNase 3 used in the MinImmune Gold kit protocol. And they all worked very, very well. No nicking. The percent recovery was between 93 and 109%. Everything worked great. We recovered most of the RNA. The protocol was very simple. So everything worked really, really well. And that's sort of the aim that I'd like to sink in for a bit, is that there's a lot of strategies for double-stranded RNA removal, and I think some of them work better than others objectively. I think a lot of them depend on the history and the perception of those tactics as well. We know that template design, optimization, reaction optimization are attractive targets for double-stranded RNA reduction. But the problem is that they aren't super consistent. They can require a lot of time to optimize, but even if they do work, they may not give you the same results every single time. And even if they gave you those better results, those results may not be good enough depending on the use that you're going to have for that RNA. The mutant T7 polymerase is also something that's come up in the last few years as being a really, really good option. And it's great for the companies that have had the resources to pour into mutational analysis. And because it's a normal IVT reaction, the attraction is there because all you have to do is kick it off. But they can be buffer specific. And once again, their lower limit is going to be something that moves around a little bit, and it may still be too high. The chromatography methods shown here as well can be varying levels of easy and costly, and varying levels of efficiency of recovery of double-stranded RNA and mRNA that you get out of them. The scaling potential varies a lot, so people usually just tend to choose stuff that's easy, right? Which is cellulose chromatography, because it's easy and cheap. But once again, if the standards for double-stranded RNA continue to be lower or higher, and you need lower double-stranded RNA, cellulose chromatography may not cut it for the future, really depending on how much more complicated mRNA therapies become and how much more rigorous the actual product is for those things. And so that's kind of why we want to bring a little bit of extra attention to using an enzyme to do the work. It benefits from being an easy protocol. It's something that you can just throw in with RNA and you purify it and you get out perfectly clean RNA. It doesn't require anything special, but it also doesn't require any upfront investment and time. And you can get to a level that is lower than almost anything that we've seen in this table, with the exception to affinity chromatography, which their level of measurement that they did for that, for instance, that Repligen paper was very, very low. And that was also just came down to how good their measurement system was. I would love to have compared by their own hands our tech and their tech to see what the exact levels were, because they did a good job of measuring it at that low level. But we are hitting a level that is much lower than mutant T7s and lower even than cellulose chromatography, in some cases reverse phase HPLC with a simple enzymatic process. And really the only thing that we ever say to do is to just test it to make sure that it doesn't cut. And so, at the risk of being a little bit repetitive, I do understand that the best case scenario is usually prevention. People would rather just not have to deal with stuff. But I think double-stranded RNA is sort of here to stay up until the point where so many iterations of maybe mutational analysis or reaction optimization are done that it just works every single time. But that day isn't here. It doesn't exist yet. And so if you have mRNA that's needed for anything that's really sensitive, you're probably going to need to clean it up. It may not pass whatever sort of measurement system you have. If you have any sort of regulations that are needed for double-stranded RNA levels, it runs a serious risk of not meeting those criterion. And if it does, you have to clean it up. And we always encourage people to do a strategy that fits their need, their capability, their scale. And honestly, there is sort of no need, capability, or scale, except in the very, very most highly structured mRNAs that just maybe don't work with mid-immune gold. But almost every single different situation using mRNA should be really well handled by mid-immune gold, just because it's cost effective, single step, nothing specialized about it, very scalable. And so we really like it and we recommend it. We think it works well. That's why we use it for everything. So I'm just the presenter, but there's a lot of people who did a lot of great work here, and including work that just goes above and beyond this presentation. Judy and Ryan, especially, they handled a lot of the actual data that was generated with Judy doing so much cell culture and transfection and qPCR, and Ryan doing a lot of immunoblots. And definitely big shout out to Ron as well, because he gave a lot of technical assistance and cloning a lot of our stuff, and definitely a shout out to Samara, who was our intern and helped us with a lot of things, and to Anu and Jen for helping to review stuff and making sure it looked, polished and crisp. And so but yeah, there's a lot of people that put work into this stuff, and they'll keep putting work into more stuff to come out of CELLSCRIPT. So thank you for your time. Questions?
Thank you, Jonathan, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type it in the Ask a Question box located on the bottom of your screen. Let's get started. Our first question asks, is there a best strategy that you would suggest for making sufficient amounts of highly pure non-immunogenic mRNA?
Best strategy may not really exist. I mean, if we're talking about a single best step to make the best mRNA possible, it comes with a lot of costs and cons, but it'd probably be something like, HPLC purification, in terms of getting the best. If cost were no object, if training were no object, and if you were cool with losing 50% of your RNA, HPLC is great. A lot of companies will provide, for instance, HPLC purification as a really costly extra step to make RNA that they provide to you. But to me, there really wouldn't be a best scenario, because that comes with so many cons. And so most anything that would need to be done would be a multi-step process. I mean, you're talking at least 2 or so strategies thrown together to produce the best possible outcome. So yeah, there's always a combination of strategies that works best, and I don't think there's any getting away from that.
Thank you for that. The next question that came in asks, do trace amounts of dsRNA have an effect on mRNA translation efficiency and innate immune activation in different cell systems?
Yes. So it's interesting because of how we can push the envelope sometimes, where we take really insensitive cell types and we'll put basically unclean samples into them, and we'll still get a good enough answer that it's totally fine. And then there will be other cell systems where you're trying to do, reprogramming from or cells that are maybe older. They've gone through a few passages. They're not quite as young and healthy, and those ones will fail pretty hard if they have even a little bit of double-stranded RNA. And we've tried it as well with doing a T7 mutant stuff in-house, where it's not as much as it usually would be, but it's not as good as it could be with, for instance, our MinImmune Gold. And those ones, they kind of just do okay, but still depends on the cell type. So short answer is yes, there's a huge effect, and it really depends on cell type, as well as the complexity of the living system that it's in. It's easy to gauge whether something works by how it works in a cell type, but then imagine putting stuff into a person and watching the number of variables increase exponentially. So yeah, it's a lot of things to consider.
Thank you for that. The next question asks, are there situations where the use of modified nucleosides during transcription, along with enzymatic dsRNA removal, may not reduce mRNA immunogenicity significantly?
I don't think so. I think that there would be combining those two strategies. It's possible, but I think combining those two strategies will get people a vast majority of the way, I would say, across multiple categories, that it would fit a lot of niches, I would say, to do those two.
Okay. How does the base composition or structure of the DNA template impact subsequent dsRNA levels in the IVT product?
Oh, huge, huge amount. I mean, we've seen that the DNA template can produce just a ridiculous amount of different stuff because there's all this extra stuff that's put into DNA templates. And unfortunately, depending on how similar they look to an initiator site, T7 polymerase, even mutants, I think can get pretty confused and start to churn out a lot of double-stranded RNA. As we've seen, if you take older school templates versus new optimized ones that have a lot of stuff shaved off, there can be a really large difference in the amount of double-stranded RNA that's produced. And that's why it really always comes down to just measuring it, because you never know and you can't ever really trust that the amount that you're going to get out is going to be low enough.
And then I think we have time for one last question. And that question asks, how does the choice of dsRNA removal method influence mRNA yield and integrity?
I think it can influence it quite a bit. For the most part, integrity shouldn't be a really big issue. Although, as I mentioned with our reverse phase HPLC, if you do HPLC, you can probably trust the integrity of your final product to be a grade higher than otherwise achievable, just because HPLC is well known to separate out stuff that's even a little truncated or short. So I think that works pretty well. Otherwise, yeah, I think the integrity shouldn't really be too much of an issue. But like stuff like yield is going to be a really big one, especially when you're talking batches of kilograms of RNA. If you're talking about losing 25, 30, 50% of that, then that can be a real big hit. And then you have to put a lot more work into producing overage to meet that. So it's nice to reduce the percent loss as best as you can. And that's why certain things I think are a little bit more attractive than other things. Like if you can do a mutant that works well, which I think I've shown that it doesn't work as well as people dream it would. But for post IVT stuff, downstream purification, affinity, and the enzymatic approach of MinImmune, for instance, would be a little bit better just because the purification doesn't lose nearly as much. So, but that's a big consideration, especially when you get up in scale.
Thank you, Jonathan. Do you have any final comments for our audience today?
Yeah, I mean, we've got the digital booth that has some cool promotional stuff to check out for CELLSCRIPT and for the MinImmune kit specifically. But really, I just thank everyone for their time, and we hope to hear from you guys. You know, I think we're always happy to have people reaching out and sending us emails and chatting with us, because you know we just love the collaboration. So, and thank you guys for providing a platform for us too.
Thank you again, Johnathan Parker, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, CELLSCRIPT, for sponsoring today's webinar. Be sure to visit CELLSCRIPT's booth in the exhibit hall for more news and information. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until October 15th, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.